Hello my dear students, I hope all of you are doing very well and you are learning every day, you are boosting up your skills and you are also becoming confident day by day after your continuous learning. My name is Abhijit Devnath, I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. So, we will be continue our learning in pharmaceutical biotechnology in unit 2. Today, we will continue our learning. From today, I am going to tell you how you can use the rDNA or recombinant DNA technology in the production of the interferon. So, I am going to tell you about these things. First of all, what is interferon? I will tell you. Then we can use interferon as a therapy. Then how you can produce the interferon by using the rDNA. Basically, this is the main topic. Then clinical cytotoxicity of interferon therapy in administration and various marketed formulation. These things are actually going to learn. Now, before starting the lecture, let me tell you there are lots of scientists around the world. Few days back, I was listening one webinar, international webinar. Some scientist she was mentioning that interferon is the first line defense of our immune system. What is immune system? That you are going to learn in our next unit, that is unit 3. For today, you just have to understand that interferon is a very important chemical substance which is falling under cytokine. It is used as a mechanism of information sharing from one cell to another. It is a communication process. You have one cell, you have another cell, both the cells are trying to communicate each other. This will become to the picture. So, interferon in short we actually write it as an INFS, a group of signaling protein or communication protein made and released by host cell in response to presence of the several viruses. You see, usually in our society, if any foreign or if any terrorist or if any antisocial person when it may enter, we always share that information with our other neighborhood, our that is people those are close to us, that yes, there is something is happening, do not that is open your door. Similarly, if your cell get infected by one virus, your cell is supposed to inform the neighborhood cells that yes, I am attacked by some virus, you should be alerted. How they will send this signal, how they will communicate, how they will inform others? The answer is interferon. This is the picture of your various kinds of interferon. So, this INF or interferon belongs to the large class of protein called cytokinin. These cytokinin molecules are used for the communication between the cells to trigger the protective defenses of our immune system that help to eradicate the pathogen. It is a very long and complicated process. This is not a simple process. Only our this interferon cannot remove or cannot destroy or cannot completely eradicate pathogen. Eradication of pathogen actually done by immune system, where we have B cells and we have T cells. Okay, we have B cells and T cells. Also, we have our that is a macrophage, dendritic cell, mast cell, etc., etc. So, it is a long process. They will be basically just inform. These are the signaling protein. They will just only inform. Okay. Now, naturally occurring proteins and glycoproteins secreted by the eukaryotic cells in response to viral infection. Also in tumor. In tumor also such kinds of interferon gamma and other things are actually released. Structurally, they are the part of the helical cytokine family, which is characterized by amino acid chain of 145 to 166 amino acid long chain. You can see it is not a very big protein. It is not a very big protein. These are 145 to 166 amino acid long protein. Okay. So, this is something about interferon. Let me tell you whatever I was telling you in my previous lecture here, that how it may that is fight against the pathogen when a virus actually entered, how they will fight against pathogen. So, you can see antiviral action of the interferon, antiviral action of the interferon. See, here is a virus and this is your entire cell. So, when the virus will enter in your cell, basically if I take the example of SARS-CoV-2 coronavirus, how coronavirus will be replicate? Very simple. Coronavirus will bind with our ACE2 receptor in our lung. Coronavirus having spike protein. So, the spike protein of coronavirus and ACE2 receptor in our lung, both the things will join together and then it will be fused and it will be enter. There are also some effect of TMPRR, SS2, S2 subunit, there are lots of story, but this is the in general this is the story how virus enter SARS-CoV-2. You may take any of the virus, when virus will enter, it will release its genomic material. If it is containing DNA, RNA, whatever the genomic material it has, it will release genomic material. After that, when it will release the genomic material, it will hijack our cell machinery, ribosome. If it is a mRNA virus, automatically that mRNA will be translated by ribosome, and what will happen? Ultimately, that ribosome will translate protein, and all these proteins, all these genomic material it will join together and new viruses will be created, and that new viruses will be infect the other cells. Usually this is happening, na? But you are lucky, because in your cell there is something called activation of interferon gene. When a virus, bacteria or pathogen may enter, your INF gene will be activated. It will produce certain protein. This is called interferon protein. This will inform the other cells that be careful, something is happening. When INF will bind its receptor to the neighborhood cell, it will signal some protein, signal its that is a nucleus. This will signal, it will create some protein, antiviral protein, it will synthesize, that antiviral protein will be destroy these viruses and virus particle will be completely destroyed. Also dendritic cell DC, they are also taking a sometimes taking a very big part, they actually show this virus particle to our T cell, that is killer T cell and other cells. So, something such kinds of things are happening, and you can see how interferon actually playing a big role. This interferon actually playing a big role in such kind of activity. Okay, because without interferon, your the cell signaling will be not happen, and virus will be continuously destroying. You may imagine, just in hypothetical scenario, you may just imagine, for example, such kind of things are actually not happening. Let me remove such kind of things are actually not happening. You can imagine what will happen. This virus entered into a new cell, from there it will create multiple new viruses. This will infect other cells, this will infect other cells, and it will again multiply, and things will be going on. That is why interferon playing a big role. Here is a detailed picture of interferon. So, when virus actually entering the antiviral action of interferon, it will be happen either single cell response or multiple cell response. In single cell response, the protein kinase which will be which actually usually inactivated, it get activate, and it may help in mRNA translation. Also oligo adenylate synthesis, which is actually inactivated, it will again become activated, and then ultimately doing a lot of process, RNA degradation will happen. In the other side, RNA editing can be possible, and in the other side your nitric acid synthesis as it will be happen. And in multicellular process, the measure histocompatibility antigen MHA, or sometimes we actually call it MHC. Yeah, MHC major histocompatibility complex will be created, and this will be virus specific cytotoxic T cell response. It will destroy the cell, it will destroy the cell which is actually affected. Such kind of things are actually happening because of the interferon. It is helping as a signal. So, INF also have various other functions like activate immune cell, natural killer cell or macrophages, whatever I was telling you. They increase the host defense defense by upregulating the antigen presentation by virtue of increasing the expression of major histocompatibility complex antigen, this one. Okay. Then certain symptoms of infection such as fever, muscle pain, flu like symptoms are actually caused by the production of interferon and other kind of cytotoxin. SARS-CoV-2, why you get fever after you are actually infected by SARS-CoV-2? The answer is very simple, overproduction of the INF or cytokine. INF actually one kind of cytokine. More than 20 distinct INF genes and proteins are actually identified in the human also, and they are typically divided among the classes like type 1, type 2, type 3. INF actually belongs to the 3 classes that are important for the fighting the viral infection and for the regulation of the immune system. So, here is the detailed classification type 1, type 2 and type 3. Among this type, within this type 1, type 2, type 3, there are sub classification. Type 1 alpha, type 1 beta, type 1 kappa, eta, okay, there are huge amounts of types. This is much more popular, okay. And yes, lambda is also here, gamma and lambda, these are much more popular. So, you can see how diverse our interferon is. Here is a very beautiful picture. So, you can see this type 1, within type 1 you have seen the alpha, okay. And also you can see in type 2 gamma and lambda. So, in alpha, the type 1 interferon, this for T cell, helper T cell, it will increase the interleukin 12R. In TC, it will activate, it will prevent the apoptosis. For B cells, it will help to differentiate the antibody production. For monocytes, it will help in for the differentiation of BAFF regulation, upregulation. For natural killer cell, it will enhance the cytotoxicity. Treg cell, it will help the separation, then T17 cell differentiation. For dendritic cells, maturation and activation, it will confirm. For other cells, it will increase the auto antigen expression. Neutrophil again, it will enhance the NET formation. Similarly, type 2 or that is a gamma interferon, what they do? They again do T helper cell 1, helper T cell differentiation, antigen presentation or antibody production, very important. For macrophage, they actually first do activation, then increase the phagocytosis, nitric oxide production, antigen that is presentation, differentiation. For dendritic cells, increase the antigen presentation. For natural killer cells, it will enhance cytotoxicity. For other cells, it will increase the autoantigen expression. Can you see how much long work they are doing? If I tell about these things, for epithelial cell, they will reduce the viral replication, also reduce the viral replication. See, this is epithelial, this is endothelial. For dendritic cells, it will help in the maturation and activation. Hepatocytes, that is hepatocytic cells, it will reduce the viral replication. Keratinocytes reduce the viral replication, and again for neutrophil, it reduces the tissue damage by inhibiting the production of the TNF and interleukin 1 beta. This is the done by lambda interferon. So, these are the entire work or entire activities what your interferons are actually doing to various human cells. Okay. If you cannot remember all those things, you will just remember these things. For example, type 1 interferon, they are doing the regulation of pathological process, induction of antiviral state, enhancement of innate immune system, facilitation of the adaptive immune system. You just remember these things, these 4 points. Okay. If you have time, then you may read the remaining things also. Now, these interferon are the first line defense against our all the viral infection. These viruses are detected by the different pattern recognition, sometimes we call it as a PAM or other things receptor, which actually all lead to the induction of the type 1 interferon. This type 1 interferon then activate various human cells, including macrophage, dendritic cells, T cells. For example, here is a picture. First, for example, proliferation of the cells are actually happening, then interferon type 1 produced by the multiple cells in presence of the PAMP or DMP. PAMP is nothing, pattern recognition. Okay. So, interferon, you can see yellow things are actually happening, it is coming. So, in third condition, induction of the IS gene antiviral state, your cell will become in antiviral state. Then in fourth stage, the recruitment of human cells will be happen. Recruitment of human cells will be happen. Human cells means T cell, B cell, macrophage, dendritic cells, etc., etc. Then dendritic cells it will differentiate in sixth stage. Then sorry, in fifth stage, in sixth stage, survival and activation of the human cells will happen. Means survival and activation means human cells, it will destroy some cells. Those cells are actually already infected, it will destroy by using cytotoxicity. Then T1 response, and later on B cell activation by antibody class switching. Means B cells are actually responsible for antibody making, they will produce antibody, huge amounts of antibody B cells will produce. So, this is in seventh stage, you can see everywhere what of the steps I have mentioned, everywhere you can see these yellow dots, okay, yellow dots you can see, yellow dots means interferon. So, this is how they are actually playing a big role. For various diseases we are actually using, for SARS-CoV-2 interferon alpha con 1, it has improved the that is a patient's health. For various other diseases we can use the interferon beta A1 alpha, beta AB, it is used to control the sclerosis, it is a autoimmune disorder. So, the treatment helps in reducing the attack of relapsing remitting multiple sclerosis. If I tell you more, interferon therapy also used in the various kinds of cancer, okay, in various kinds of cancer actually using, such as leukemia and other kinds of lymphoma and other kinds of diseases. Patients with melanomas also receive the recombinant TNF alpha B2. Both hepatitis B and C are treated with the interferon alpha. It is often used with the combination of the antiviral drugs. If I say formulation, several types of interferon actually used. First approved was in the 1986. In January 2021, FDA has approved PG, that is pegylated interferon alpha in US. Interferon containing regimes may also include protease inhibitor such as bezafibrate and tenofovir. These things are actually some antiviral drug, where you will be observing it was indexed with VIR. It was the name of the drug as indexed with VIR, you should understand it is a antiviral drug. You can see here, VIR means these are the antiviral drug. So, interferon actually used among with them. There are also interferon including drugs, notably telunarone, that shows to be effective in the against Ebola virus. Can you see how much popular our interferon is? Now, how will produce? Production is very easy. If you understood my previous lecture of rDNA, it will be very easy for you. So, the complementary DNA to be synthesized from mRNA of a specific interferon, and this to be inserted into a vector which is introduced into E. coli. See, complementary DNA, whose DNA is actually responsible for interferon development, actually synthesize that one. Then you are going to insert into the vector, that is plasmid. Now, how you will do? How you will this insert the foreign DNA into plasmid by using these techniques, either transformation, either electroporation, either protoplast infusion or microinjection. Okay, microinjection you can see this is the process by using you can do. What is the entire picture? Entire picture is very simple. You have bacterial cell in stage 1, you have a bacterial cell what actually enzymatically digested, which is actually producing the protoplast, and then you this both the things are actually you have joined together, protoplast fuse, and then you got the recombinant cell, and this recombinant cell will grow to the new cell, and that thing will produce your interferon. Okay. And interferon can be isolated from the culture medium after production. After when you will express interferon, you will be able to take out. So, interferon alpha 2, interferon alpha 3 genes are actually digested by actually restriction enzyme. So, when it will digest, you can collect that thing, and the resulting fragment will be ligated to generate the hybrid gene. For example, this is your interferon alpha 1 gene. You have used some restriction enzyme. What is restriction enzyme? In my previous lecture I have told you that molecular scissor, it will cut the DNA, like this. okay. And you have a few fragment, and that thing you are actually going to add it with your gene of interest. Both the things will join, and then you will be having the hybrid gene, and you can implant those things into the E. coli. E. coli you can use, and then this hybrid gene will be produce your this thing called interferon, which you can isolate from the culture medium. Okay. So, the process is very simple, but I have told you lots of positive things about interferon, and I have told you how you can use interferon in various viral therapies. You also have seen interferon has a very big role in cell signaling, especially when viruses are actually entering. But there are some issues. There are some clinical toxicity of interferon therapy administration. You have to take care of those things. During clinical trial, you have to you can check how much you can minimize, but in present day scenario there are lots of clinical cytotoxicity have been reported to us. For example, constitutional cytotoxicity, hematological cytotoxicity, then organ toxicity, then neuro psychiatric toxicities, then endocrine and metabolic toxicities, also potential drug interaction. These are the major toxicity or cytotoxicity you can say present day we are actually facing. So, when you will do some project, some research work on this area, you have to take care of these things. You may try your best so that you can minimize their impact, but what I believe it will be not possible, because still the date we are actually depending on our that means human induced or whatever the interferons are present in our body, we are relying on them. Okay. So, if you are able to modify some of the structure, some of the motif, some of the amino acids, and if you may able to stop their off target binding, if you may stop interference of target binding, then it will work, because whatever the toxicity I have mentioned here, all the toxicities are happening because of the off target binding. Means the interferon actually it is going to bind other human protein where it is not supposed to bind. That is why you are observing these things. Now, let me tell you few of the marketed formulation. You can see these are the picture of few marketed formulation. You can see PEG interferon alpha 2, okay. And you can see another one interferon alpha 2. All of you are I hope you are aware about the brand name and generic name. You should be careful about these things, brand name and generic name. These are the two other pictures of interferon whatever actually given. You can see alpha 2A, mostly alpha 2A actually used. You can see another of interferon. I think they have not yeah, here is alpha 2A. This is developed by Roche. Roche is the most famous that is a biotech company, okay. And this is actually developed by Genentech. This is actually developed by again Roche. It is the old one, what I believe. This is the old picture and this is the new picture of Roche. It is a very common popular company in our this biotech field. So, we are actually on the ending of our lecture. So, whatever the things today I have learned, today I have told you about the introduction to interferon. You have seen interferon therapy, you also have seen interferon production by using rDNA. Then how clinical cytotoxicity or toxicity actually happening after interferon administration, and then you have seen the various marketed formulation. So, in my next lecture I will tell you more applications of recombinant DNA technology in vaccine production. I will tell you hepatitis vaccine production, how you can use recombinant DNA technology for hepatitis DNA vaccine production. So, stay with me, listen my next lecture. This lecture going to be very interesting, because lots of insights I am going to tell you in the next lecture. The system will be completely same. there will be one plasmid, there will be one gene of interest, you are going to fuse both the things and you are going to get a you are going to insert you are going to that means place that entire complex into a E. coli like bacteria. When it will express that protein, for example, this hepatitis B vaccine. So, thank you all of you for joining with me, for listening all the entire lecture with a great patience. So, I hope all of you have learned our this topic in a great detail, and this topic will help you in your future study and research. So, God bless you all. We may meet in our next lecture. All the best.